Yeah, so we've been talking, we ended up last time, I believe, talking about these two terms, maintenance rehearsal, elaborative rehearsal, which I will go over those in a second. Yeah, so that's just to kind of refresh ourselves. We also have been talking about this levels of processing study, like this one right here. People read lists of words, and they're either paying attention to whether the letters in that list are capital or lowercase. Another group is paying attention to whether each word in the list rhymes with some certain target word. And then a final group of people is asked to look at each word and imagine and determine whether the word you're looking at fits in a list, or sorry, if it fits in a sentence. So the examples are here. Is the word in capital letters? That's like the case condition, upper lower case. Does the word rhyme with the word wait? That's the phonemic condition. And then we have the sentence condition. Would the word fit with the sentence? He met a blank in the street. So this is like semantic or sentence condition. And then they looked at people's ability to remember these words later on. And they found that the people that had originally encoded the words in terms of meaning, the people doing the sentence task, they had high recognition, high ability to remember words that they had seen in the list originally, whereas people that were just paying attention to the case of those letters in the list, their memory was poor. So that's just another finding in this chapter to kind of establish what we've been talking about last time. So related to that study then is these two terms right here, maintenance rehearsal versus elaborative rehearsal. I believe we did go over these last time. Maintenance rehearsal is a process which appears to rely primarily on short-term memory. So here's kind of a schematic diagram of these different memory stores. And in short-term memory, there's this kind of self-referential arrow or rehearsal right here, where you can kind of say information to yourself in kind of a superficial level, and just repeating something verbally to yourself kind of over and over and over again. And the idea is that eventually that information can be transferred, and that's words even written here in this diagram. When you use rehearsal, that information can be eventually transferred over to long-term memory. But in the previous study, there was pretty clearly a benefit of elaborative rehearsal. So once again, in the sentence condition, each word you're evaluating the meaning of that word. And so that's a type of elaborative rehearsal when you're using meaning to help encode words into storage. The question in the previous studies, the levels of processing study, is do you get a benefit in memory when you use an elaborative type of encoding strategy like the sentence condition? The answer is yes, compared to a more superficial type of encoding strategy like is the word written in upper or lowercase level. So the sentence condition in the previous study would be an example of elaborative rehearsal. You're using the meaning of things, and that kind of gives you a benefit of remembering those things later on. If you think about the meaning of something, it helps you to remember that thing later on. This elaborative rehearsal type of thing, in a sense, is using your long-term memory to help with retrieval and transfer. So it's kind of this bidirectional process when we're talking about elaborative rehearsal. You're using something that you already know, the meaning of certain words, and the meaning of words in your long-term memory that's stored here. And then when you see those words, you're holding that information in your short-term memory while you're simultaneously thinking about the meaning of that word, which is in long-term memory. You're creating a pathway, in theory, between your short and long-term memories, which then strengthens the connection you have between short and long-term memory, which is the explanation for why you are apparently better able to remember things after using elaborative rehearsal or elaborative encoding relative to a more superficial type of encoding like the case condition or the phonemic condition. So any questions about maintenance rehearsal and how it's different than elaborative rehearsal, and how the concept of elaborative rehearsal connects to this levels of processing study right here? Questions, comments? Moving on. So another example of elaborative, I really like the term elaborative encoding better, but elaborative rehearsal is used interchangeably. So elaborative rehearsal or elaborative encoding. Elaborative encoding I actually prefer, even though it says rehearsal here on the slides. So here's some examples of, make sure I'm not losing you people, okay. Here's some examples of mnemonic devices, which are further examples of elaborative encoding. So the method of loci. This is where you use like a story in your head where you picture locations of different items in a list. And you use those locations to help you remember the items in the list. So as a researcher, I give you a list of words that I want you to remember. And this method is if for each item in the list, you kind of travel through some location that you know, some series of locations that you know, and at various landmarks along the way, you imagine yourself seeing that each item at that location. So like I think in the book, like the first item would be like banana. And so if we're at Fraser Hall at KU and we're entering the building on the bottom floor, you go up those stairs that are outside and I imagine a banana right there on that little ledge thing, the little banister thing as you're going up those stairs outside Fraser Hall. And then inside you open those doors and then you get to the little like area where you can either go up the stairs or you can either go into the hall and then you see another object there like a suitcase. And so you know, banana is the first item in the list. The word suitcase is the second item in the list. You have a list of like 10 items, and for each item, you imagine it at some known location, and then you kind of travel along the path of those locations and in your mind, and at various landmarks, you imagine seeing the item, and then in theory or the idea is that this should help you later on to remember the words that are on the list. This would be better presumably than using like a maintenance rehearsal type of strategy. In maintenance rehearsal, I give you a list of words to remember, and the way you remember it is just like saying those words in your head over and over and over again. This type of elaborative rehearsal or elaborative encoding, you're using locations, method of loci, you're using the locations of things. You're connecting that to each individual item. You go through these different locations in a sequential order, and this should help you. This type of elaborative rehearsal, elaborative encoding, should help you remember those items in a list better than something like maintenance encoding. Another example is the story mnemonic, which is really kind of like a type, or it's more like like the method of loci is like a specific type of story mnemonic. So method of loci is a specific type of story mnemonic. And the thing that makes the method of loci unique is that you're specifically relying on locations of things. You're creating a narrative in the method of loci, which is based on locations of things. So you're creating a narrative, a sequence of events based on different possible locations of things and imagining an item for each one. Method of loci. Story mnemonic is just a more general idea, just a more general type of elaborative rehearsal or a more general type of mnemonic device where you just basically tell a story about a list of items. You might not necessarily be traveling through a series of locations and seeing each of those items, but you make up a story, and that story helps you to remember the items in the list. The book mentioned that this might be especially helpful for kids because kids find memory tasks boring. I mean, so do adults, but you could make a task more fun for kids, a memory task, if you incorporate it into this type of story format. And tell a narrative, and then that can help kids remember. So it might be particularly beneficial, the story mnemonic, for kids because it motivates them to learn or to remember, when otherwise just doing maintenance rehearsal or something like that in kids, they are very inattentive to those types of strategies. They're more attentive to like story type of strategies because they can be more interesting. It's more fun for them. Any questions about mnemonic devices, elaborative encoding? Okay, so we've been talking about elaborative encoding, and particularly with the method of loci. That was an example in which you were creating memories of things, declarative memories, memories of that you can verbalize. These are the words that were in the list. But method of loci is an interesting type of elaborative encoding because it's tied to contextual experience. It's your ability to connect objects, items, to a context in which the item was experienced, or at least you thought about experiencing the item. And the regions of the brain that appear to contribute to this type of elaborative encoding have been well studied. We've actually talked about it a little bit before, but now we're going to go into more detail about the medial temporal lobe system. I don't think it says here, which is good. I want to jump down here. HM had parts of his medial temporal lobe removed, and he showed a declarative memory impairment, but his procedural memory was intact. What was the specific region of HM's medial temporal lobe that was removed? Does anyone remember? There are several regions that were removed, but there's one region of the medial temporal lobe that's generally been considered most important for the encoding of new long-term declarative memories. Does anyone remember the name of that brain region?
Is it the hippocampus?
Yes, it is the hippocampus. That is the region in the medial temporal lobe which has been most often connected to the encoding of long-term declarative memories. Remember, that was what HM had his problems with. His procedural memory, his performance in that mirror drawing task was preserved, but his long-term declarative memory was impaired. Turning on the light here. And so we're going to be paying more attention to the whole medial temporal lobe system, but the hippocampus, which is kind of the heart at this. It's important, particularly for episodic memories that involve where things occur, where either me or whether objects occur relative to other objects or relative to a scene. It's connecting objects to some type of contextual experience. And that contextual experience mostly is where objects are relative to other objects, or where objects are in a given scene or a given room. And we'll talk a little bit more about that, about this role of the hippocampus in the next few slides. Yeah, that's actually it for the slide. Yes, so let's go to talking about some of the, remind us where this region of the brain is. Hippocampus is right here in the medial temporal lobe. We will be talking a little bit about some of these other regions, so I'm not going to require you to know anatomically these different regions of the medial temporal lobe, so you won't have to label these. Though I think I'll probably likely ask a couple questions about the function of 4 different regions here. I'm gonna move, The 4 different regions which I'm going to care about you knowing the function about is hippocampus, which is the blue region right here, pararhinal cortex, which is right here, entorhinal cortex right here, and parahippocampal cortex. Those are 4 regions, all in the medial temporal lobe. They all work together for encoding long-term declarative memory, particularly, apparently, episodic memories, memories from a first-person perspective. And so we'll be talking about these. Once again, anatomy I'm not going to be too concerned about, though I am interested in you knowing what I'm going to show you about the function of these brain regions in the upcoming slides. So here is a kind of complicated diagram, but it's giving some important information about these 4 different regions of the brain and of the medial temporal lobe, and their potential functions for the forming of long-term declarative memories. And so you can see there's kind of these 2 input streams. There's a what input stream and there's a where input stream. And according to this diagram, the what input stream involves the PRC, the pararhinal cortex. Pararhinal cortex, I showed you that in the previous slide. The where stream, the brain region involved here, is the parahippocampal cortex. And then these both converge here, EC, which stands for entorhinal cortex. So we have these 2 different regions that are kind of converging to one. I want to go back to our brain here. So the pararhinal cortex, that sends signals to the entorhinal cortex. And the parahippocampal cortex right here, that also sends signals into the entorhinal. So we have kind of these 2 brain regions located on each side. The entorhinal cortex is in the middle, parietal cortex is on one side, parahippocampal cortex is on the other. And these two regions kind of feed in. They both input signals into the entorhinal cortex, which is in between them. The parietal cortex gives what information? So this is telling you, what are the items that I'm remembering? What are the objects that are relevant that I'm trying to encode information about? Item or what information, what the objects are. The where part of this, the posterior parahippocampal cortex, you can think of this as kind of providing spatial context. So where objects are relative to each other and where objects are relative to a given room, though it does get a little bit more complicated. There are other type of contexts that appear to be represented here. So spatial context is an important one, but other type of contexts are important here. An example, which I won't elaborate on too much, but for instance, you have a memory of a person, and part of the context would be the color of the room that you met that person at. Or part of the context might be what the person was wearing when you met them. So that would fall under this category of what's going on in the parahippocampal cortex. Where is the main type of context that this region of the brain is interested in? But other type of context too, like the color of a room would be like another example. The color of the environment that you are in when something happens would be another example, which I've seen some research supporting a role. So context in general, though, where is the particularly important part of this brain region right here. Both of these brain regions, parahippocampal cortex feeds into the entorhinal cortex, which is in the middle. The parietal cortex feeds into the entorhinal cortex, which is in the middle. And then both of those the entorhinal cortex then sends both of those types of information, the object information and the contextual information. The EC, the entorhinal cortex, sends that information up into here, which is the hippocampus. So the hippocampus is composed of these 4 subregions. You don't need to know these at all. My cognitive neuro class, I might make them know a couple of these, but for you guys' class, you definitely don't need to know these different subregions. It's the subiculum, dentate gyrus, CA1, CA3, just so if you want to know how these are pronounced. These are 4 subregions of the hippocampus. The EC, the entorhinal cortex, sends information into the hippocampus. So here's kind of another, this is kind of, well, for one thing, there's another picture of the hippocampus and some surrounding regions here. This is an actual brain with some of these regions labeled anatomically. But this slide also gives you some information about the functions of these different regions. It's kind of reiterate some of the stuff from the last slide regarding the functions of these different regions. So we know that in general from the lesion studies, the hippocampus is necessary for the encoding of long-term declarative memories. When HM's hippocampus was lesioned, when it was removed, he was no longer able to form new long-term declarative memories. The entorhinal cortex EC, I mentioned, that receives those 2 inputs and then sends the signal up into the hippocampus. You can actually see the entorhinal cortex in the stained brain right here. There's the EC, and you can see that there's an arrow going from EC in to the hippocampus right here. So this whole thing up here is the hippocampal formation, dentate gyrus, CA1, CA2, CA3. Oh yeah, the subiculum is up here. So these are different parts of the hippocampus right here. The entorhinal cortex is sending the inputs into the hippocampus. The entorhinal cortex itself is receiving inputs from those 2 different regions. The parietal cortex, it inputs item information, object information, information about what into the entorhinal cortex. And this is a part of the brain which, like when the objects are familiar, this part of the brain is important. This is part of the brain which is activated strongly when you're recognizing a familiar object. And then the parahippocampal area, as I mentioned this before, it also inputs information into the entorhinal cortex. But whereas parietal cortex inputs item information, what information, parahippocampal cortex inputs contextual information. So oftentimes that's studied in terms of information about where things are located, but there are other types of contextual information too. I mentioned color earlier, the color of a room. Yeah, that was probably the one example I'll stick with. I want to also point out some interesting things about the hippocampus here is that it's a loop. You actually see here, this has come from a recent article in Current Biology. It's a pretty prestigious journal. And they talk about this in the little figure legend or the caption down here. They talk about a trisynaptic loop. And that loop refers to the fact that there's kind of this cycle of things that are going on. Information goes from parietal cortex, parahippocampal cortex to the entorhinal cortex. And then the entorhinal cortex sends the information up into the hippocampus. But then the hippocampus is sending information back into the entorhinal cortex, which is then presumably influencing the way information is filtered back into the hippocampus. So there's definitely kind of a bidirectional thing going on. Information is going into the hippocampus from the EC, but then information from the hippocampus is going back into the EC, back into the entorhinal cortex. That's that loop which is being talked about. So some of the evidence that these regions of the brain, the hippocampus and the entorhinal cortex, these regions of the medial temporal lobe, some of the evidence that these regions are important for for using contextual information, particularly spatial information, and forming long-term memories, comes from this research on these specific types of neurons that are found in the medial temporal lobe, especially in the hippocampus, also in the entorhinal cortex, are these where these two regions are found primarily. That there's two types of neurons, one's called place cell and one called grid cells. I will describe place cells first. These are ones which are found in the hippocampus, although you don't need to know where place cells are found, other than you need to know place cells are found in the medial temporal lobe. You don't need to know that they're specifically found in the hippocampus, though they are found specifically in the hippocampus. So this is a diagram right here, this A diagram of a place cell, a single place cell. And what these little black lines, this is like a rat, and these little black lines are where a rat is moving around inside a room. So the rat's moving all over the place. And they're measuring, while the rat's moving throughout this room, they're measuring the activity of a single neuron, one neuron in this rat's medial temporal lobe. And they're looking, when does this neuron fire action potentials? When does this neuron become electrically active? And for place cells, a specific type of neuron in the medial temporal lobe, they find that this place cell, this specific place cell, fires a lot of action potentials when the rat is in this part of the room. But when the rat's in the other parts, like when the rat's in this corner, or this corner, or this corner, or this corner over here, or even this part over here, the rat, this one place cell, is not firing action potentials. But when the rat moves into this part of the room right here, where these red splotches are, then that one neuron fires a lot of action potential. And there's multiple place cells in your medial temporal lobe. Different place cells would become activated when the rat was standing over here. A different place cell would become activated when the rat's over here. Basically, different place cells represent you as the person, as the person who's forming a memory. It represents your location within some type of spatial context. In this case, it's like a square room, apparently, or a cage maybe for a rat. That's a place cell. Each place cell fires action potentials when you, the animal, you, the person, or the person, are in a specific location in a context. Grid cells are a little bit more complicated. Grid cells, which are located primarily in the entorhinal cortex, they have a grid-like pattern of firing. And you can see that grid-like pattern right here. The grid is the fact that there's not, when the rat's standing right here, so this is all right here, all these red splotches are the activation, once again, of a single neuron, but now it's a grid cell, a grid neuron. When the rat is standing right here, this grid neuron does not fire action potentials. But when the rat moves and is standing in this part of the room against the wall here, it is firing action potentials. The rat moves a little bit more. This same neuron stops firing action potentials. It moves a little bit more over here. It starts firing action potentials again. And that's the grid, or the lattice-like structure of grid cells, where a given grid cell, it doesn't just respond to one single location like place cells do. It responds to a grid-like set of locations, once again, though, within a specific context. And one theory as to what's going on. So it's not cognitively, it's not exactly clear why these cells are organized or they respond in a grid-like fashion. But one theory is that it has to do with recurrent processing. Once again, that loop. Information leaves EC, which is where these grid cells are. It leaves ECs for the hippocampus, and then information from the hippocampus is sent back to the interrietal cortex. There's that loop there. And that might explain why the interrietal cortex kind of has this intricate, complex way of representing space, because it's kind of representing input and output at the same time. So yeah, this is some pretty advanced, this is actually some pretty advanced cognitive neuroscience, but it's important for understanding some of the purely cognitive things which are going on in the hippocampus. So I want to give you, is there any questions or comments about grid cells? People still alive out there? All right, no questions. So moving on to consolidation theory, which is another neural theory, but there's just some really cool cognitive stuff that goes along with this cognitive theory, or this neural theory. So the hippocampus we've already talked about. We know that it's important for the encoding of long-term declarative memories. But it also turns out that the hippocampus is important for retrieval of newly formed memories. So when if I meet someone today and I meet them, shake their hand, they tell me their name, that information, I can remember that the next day, hopefully. And the next day I meet that person again and I have to retrieve their name from long-term memory. If you were to measure my brain's response, there's likely to be a high amount of activity in my hippocampus that day after I encoded that long-term memory about the person's name. And that's because when you have a freshly formed long-term memory, the hippocampus is important for retrieving that newly formed memory. However, the hippocampus involvement in retrieval declines the further along in time you go relative to the original formation of that long-term memory. So if I meet a person and then I run into them 30 days later, and then I have to remember their name again, and then you look at which regions of my brain are most activated when I'm retrieving the memory about this person's name, you're going to most likely see less activity in my hippocampus 30 days later than you would 1 day later when I'm trying to retrieve the person's name. And the explanation for this is consolidation. The idea of consolidation is that a different set of brain regions, a different separate neurocognitive mechanism is important for retrieval of information 30 days or a long time after encoding a long-term declarative memory compared to a short amount of time, 1 or maybe 2 days after forming long-term declarative memory. In the brain, the hippocampus is important for retrieval after a freshly formed memory. But if you look at what brain regions are important 30 days later, after the memory's been formed for a long time, it tends to be what I'll just kind of vaguely call outer cortical regions. The hippocampus is medial temporal lobe. It's inside, kind of in the middle part of the brain, and it's relatively down low. When you're retrieving a long-term memory that's like 30 days old, you're going to see more activation on like more lateral, more brain regions towards the side of the brain, and outer cortical regions like toward the surface of the brain, rather than medial regions like the hippocampus. So retrieval is the same cognitive process in a sense, but when the memory is fresh, the hippocampus is the brain region which is mostly involved with retrieval. When the memory is a little bit old, a different set of brain regions, these outer cortical regions, are involved with retrieval. That's consolidation. Consolidation is the idea that with time, a new set of brain regions becomes dominant in the retrieval of a given memory. So the way that this has been studied in laboratories, or one popular way, is with fear conditioning, where the rat learns an association between an object, a thing, in this case it would be a shock that the animal receives, and the occurrence of this shock, the experience of the shock in a specific contextual location, like cage number 1 compared to cage number 2 compared to a third cage. So you have 3 different cages. One of those cages, when the rat is placed into it, they get a shock every time. The other 2 cages, when the rat is placed in the cage, the rat does not get a shock. What the rat usually does, if their hippocampus and their brain is working in general to form long-term declarative memories of this, bad experience, receiving shock, which causes them to experience fear. The rat, if they remember this fear experience, will freeze when placed in that environment again. So when the rat is placed in cage 1, presumably it's going to freeze because it had been shocked there before. And if it remembers, oh, when I was in this context, when I was in this environment previously, something bad happened, the rat's tendency is to freeze. The reason the rats do that, presumably, is because usually when they feel comfortable, they'll explore a little bit. They'll kind of get a feel for the place. But if they've had a bad experience, they're going to be less likely to engage in exploratory behavior because exploratory behavior feels risky to them. So if they remember this is a bad place, the thing that they're likely to do is freeze. And so, yeah, I know this figure is a little bit cloudy, but it illustrates well this idea of consolidation and how consolidation changes from relying strongly on the hippocampus for day 1, or day 1 after a memory, a freshly formed memory, versus, in this case, I guess it's 28 days later in the second condition. So 1 day after a memory is formed versus 28. So the memory that the rats are forming in the study is fear conditioning. All the rats learn to associate a specific environment with the shock. And the rats are in one of a few different conditions. So the saline, this is like the placebo or control condition. So what they do in this condition, the rat is placed in the environment which they've had a shock in before, so they should be scared of this environment, and they just inject saline into their hippocampus, which basically does nothing. It's, saline doesn't change anything important in the hippocampus. So it's like the placebo or the control condition. Basically, they're getting an injection of something, but it's a weak substance, saline, which doesn't do anything to their brain. That's like the control condition. Compared to this CNQX, I actually don't know what that acronym means, but I do know that this is a substance that is thought to turn off the hippocampus. It makes it so the hippocampus cannot be used. And so if the hippocampus is working, if they're able to form the association between this object, the shock, and the contextual environment in which they receive the shock, if the hippocampus is working, they should show a lot of freezing behavior. The large percentage of the time when they're in the cage, they should be still. And they do that in the saline condition. The saline condition, their hippocampus is intact. But when their hippocampus is slightly impaired by this chemical which is injected into it, you see that the freezing behavior decreases. That's because when their hippocampus on day 1, this is 1 day after they learned about the shock in this particular environment. When the hippocampus is damaged 1 day later, the hippocampus should be involved with the retrieval of this memory, but the hippocampus is damaged, and so they have trouble retrieving that information that they had just acquired the day before because the hippocampus is damaged. That's day 1, or when the memory was the fear memory was freshly formed. Damage to the hippocampus makes it difficult to retrieve that memory, which results in less freezing behavior. 28 days, though, this is where some really interesting findings occur. In general, you can see that 28 days later, they show less freezing behavior, which makes sense because it's been 28 days, so they've forgotten to some extent. The memory is less strong in their mind, so you can see these bars are like the saline bar right here is smaller than the saline bar here. This is just forgetting. 28 days later, the rats are placed in that same environment again, and even if they've injected with saline, which keeps their hippocampus preserved, they show slightly less freezing behavior, which is probably just because of normal forgetting that occurs 28 days later. What's interesting, though, is that if you look here in the CNQX condition, this is once again when they're injected with this chemical into their hippocampus, which impairs the hippocampus, it damages it, makes it so that you can't use it. You notice, though, that there's really not that significant of a detriment of their freezing behavior. They still freeze about the same amount as they did when they received saline. After day 1, there's a big drop off. They're really good with saline, but there's a big drop off when their hippocampus is removed. But on day 28, they're okay with saline, but you notice they're about the same amount of okay, even when their hippocampus is damaged. So what this suggests is that you need your hippocampus to retrieve the freshly formed memory but you do not need your hippocampus to retrieve that memory after 28 days later. Presumably, the reason why the rats here in the CNQX condition are still showing a decent amount of freezing is because the memory that they formed, the shock occurs in cage number 1. That memory no longer resides or requires the hippocampus to retrieve it. It's now relying on outer cortical regions. Those outer cortical regions have not been damaged by the CNQX. And so the animal still remembers, oh, this is a place, this cage has shocked me before. And so the animal shows freezing behavior. Any questions so far about consolidation theory? I have a little bit more to say, but any questions so far about it? So here is a neat kind of idea about a theory of how consolidation theory works in the brain. You don't need to pay attention to this figure right here, but I do want you to pay attention to these two figures right here. This big rectangle right here is the hippocampus. Same thing again, hippocampus. And these over here, it calls them cortical modules, but just imagine that these are like the cortical regions of the brain, the outer cortical regions. So the hippocampus represented down here, the outer cortical regions are represented over here. And these red lines basically represent pathways, interactions between these different brain regions when you are retrieving a memory. In this one right here, it says hours. This means this is what the brain is thought to look like when you're retrieving a freshly formed long-term memory. So within like a day or two of forming a long-term memory, the hippocampus is highly active. And what the hippocampus is doing, it's sometimes thought of as like a conductor, it's sending signals to all of these other cortical regions of the brain. The hippocampus is kind of the heart of this process, and it's sending signals to these other cortical regions of the brain, the ones that are outer cortical regions of the brain. And the process of retrieval is kind of spurred, or it's conducted. The main organizer is the hippocampus, and it does so by recruiting all of these outer cortical regions of the brain. This is when a memory is fresh. But when the memory is relatively old, so here it says weeks, so this is weeks after you formed a memory. The hippocampus, you'll notice, as I've suggested in the previous slides, the hippocampus is not as active anymore. But what is active is the interactions among these same outer cortical regions. And you'll notice when the memory is fresh, there's no connections among the outer cortical modules, the cortical regions here. There's no red lines between these small rectangles right here. What this represents is that when a memory is freshly formed, there's no connections between the cortical modules. There's no connections among all of the different outer cortical regions. And so when you're retrieving a freshly made memory, you need the hippocampus to activate all of those outer cortical regions together. But eventually, because of consolidation, connections form between these outer cortical regions so that 28 days later, several weeks later, the hippocampus is not necessary. Connections form by themselves among these cortical regions so that when you're trying to retrieve the memory, these cortical regions can do it by themselves. They don't need the hippocampus to conduct things. They don't need the hippocampus to organize things. Yes, so that's consolidation. Still no questions or comments about consolidation? All right. Just a couple more things, and then we'll take a break for this chapter. So these are just various facts about long-term memory. There's been a lot of themes in this chapter, but these kind of are interesting facts about long-term memory, and they didn't really fit in with some of the bigger picture themes. So I'm kind of just putting them in as various interesting facts about long-term memory here at the end of the chapter. So the first kind of fact is the idea that distributed practice leads to better retrieval of long-term memory compared to massed practice. So this is actually what I did my science fair project on in Mrs. Toussaint's 7th grade, or was it 8th grade, science class back in Indiana. I didn't know of these terms, massed and distributed practice, but this was definitely my science fair project that year in junior high. And so let's kind of look at these terms. Massed is when you study for like 4 hours, a long time, in one study session. relative to distributed learning, where you might still study 4 hours, but you do so in several sessions, and each of those sessions is shorter. It's not one session of 4 hours. It's a bunch of sessions that are each shorter than 4 hours. And the question is which of these is better for later? You're studying one way versus the other. Which one is better for your ability to later retrieve the information that you are studying? And I can say, I know that sometimes in college, I feel like I probably use massed studying more than I use distributed, but that probably was not a good idea, because distributed studying appears to be better. That is, your memory of things, your ability to retrieve long-term memories is better with distributed studying, distributed practice. The primary explanation that I've seen is mental tiredness. When you're studying for 4 hours straight, like you do in massed practice, it's kind of fatiguing, especially to your attention parts of your mind. It's hard to kind of stay focused on any task for a long period of time. And kind of basically another way of saying that, you get mentally tired. And so your brain becomes less efficient when you're doing massed encoding, massed practice, which is the primary explanation for why distributed encoding or distributed practice is better. So that's fact one. Distributed practice is better than mass practice. We'll also talk about these two other facts down here: encoding, specificity. So here's kind of the famous study about encoding specificity. You're learning a list of words, and that's the learning context. And you either learn on land or you learn underwater. So you're memorizing a list of words in one of these two contexts, one of these two situations. And then you later on are tested on the same things that you had learned earlier. So like a list of words or some type of information you have to learn about. You're then later on tested for that same information. And you either are tested in the same context in which that information was originally learned, or in a different context. So let's kind of break things down. The dependent variable here is accuracy, or the amount of words you're able to successfully remember. When you learn on land, this column, you are better. Your performance is better when you're tested on land as well. When you learn on land, but you're tested underwater, your performance is bad. But it's not that just being tested underwater is inherently hard. It probably is a little bit harder. But if you actually look here, when you learn underwater, your performance is actually better when you are later tested underwater as well. If you learn the list underwater, but then you're tested later on land, which is what the red box is, you're later tested on land, your performance is actually bad. So this illustrates the encoding specificity quite clearly. If you want to have success retrieving information, you are going to have the most success if you are encoding that information and retrieving it in the same context. And then finally, state-dependent learning, which is shown, described here. So yeah, this is a controversial one, because students kind of hear about it and they're like, oh, this means that if I drink Friday night when I'm studying, that I should get drunk before my test, and then that will be a good idea, which isn't exactly true. But there's some interesting evidence, interesting results here that we'll talk about. Once again, this is state-dependent learning. So you encode like a set of words, or you're learning some type of information in one of two situations. You're either sober or you're intoxicated. So you're either, drunk or something, or on some other drug, versus completely sober right here. And then that's just the learning, that's the encoding phase. And they'll later test your ability to remember the information that you previously studied during encoding. And then you'll either be tested when you are sober again, or when you are not sober again. Or so you'll either, some people were encoded the words when they were sober and then they were tested when they were sober. Some people encoded the words when they were sober, but then were tested when they were intoxicated. Some people encoded the words when they were intoxicated, but then were tested when they were still intoxicated again later on. Some people encoded the words when they were intoxicated, but were later tested when they were sober. And they want to see how does your state in encoding, how does that interact with your state at retrieval, whether you're sober or intoxicated retrieval. So first thing, you notice that when you're sober, you are most accurate in this task. When you're sober at encoding, you are going to have more success in this task when you are also sober at retrieval. And you can see that by the relative position of the dark circle here relative to the light circle. This one's taller than this one. So if you're sober at encoding, the best strategy, which doesn't seem surprising to me, is to also be sober at retrieval. In general, sober is the best strategy. The interesting finding is over here. When you're intoxicated during studying, once you are intoxicated at studying, it actually makes some sense that you will actually be slightly better if you actually retrieve that same information while you are also intoxicated. So if you do drink during studying, then it actually would be slightly advantageous, at least in theory, according to these results right here, to drink before your test, drink before your retrieval. Now, I say that, and it is true, your performance is relatively better here if you are intoxicated relative to if you are sober during retrieval here. But as you can see, the intoxicated condition, there's clearly a main effect of state of encoding, where if you're sober in general, if you're sober at encoding, you're definitely going to be pretty darn good during when you retrieve stuff. I'm actually surprised at how good people are. The white circle right here, these are people that are intoxicated when they're being tested, and their performance is actually still pretty darn good. People, though, if you are intoxicated at all during encoding, you can see your performance is going to be bad, especially, in fact, if you're sober at retrieval. So don't think that you can get away with, drinking during studying. It's a terrible idea. Obviously, it has very large impairments on your later retrieval. But if you're going to drink during encoding, it actually would be your best strategy to drink during retrieval as well. So that's where we'll take first break. I'll give you 10 minutes. So it's 8 o'clock right in the nose. So let's come back at 8:10, and then we'll start the second half of class today.